I, I would like to start by two comments. First of all, we moved our institute to a new building in 2018. So I know how hard it is and how frightening this is for labs to move from place to place. I really congratulate you for this new building. I know also how much this improved the quality of the space for many of these labs. So I wish you really a start of a long and very productive period in this new building. The second comment I want to make is that I'm really surprised at how many people remained to listen to this last session. So thank for all of you who are here. I want to start by presenting the people who really did the work. I'll talk about the contribution of each one of them separately. Anna and Alex are PhD students, and Johannes and Maciek are postdocs in my lab. Okay, so I did, Dan mentioned the work that I did about stimulus-specific adaptation and surprise, predictive coding, and so on. I did all of that in anesthetized animals, and anesthetized animals are great. Okay, much better, much simpler to work with than awake animals, but with time, the requirement to work with, to see what's going on in the awake brain became overwhelming, and so I wanted to study in the last 7 years or so. We started studying behaving animals and obviously auditory guided behavior. Now, my problem was at the time that we started to do these studies that I found most studies of sound responses in awake animal quite boring. And the reason is that in most of these studies, you get two highly discriminable stimuli, and what the animal has to do is to go left to right. So that's simple and not very sophisticated, and it's not really addressing what auditory cortex is doing. And so what we did, I mean, our solution for that was to go and build a bit naively an environment that would allow more complex behaviors driven by sound. The goal, not very well-defined goal that we had, was to study the coding of task-relevant versus task-non-relevant sounds. And as you will see, what the really interesting stuff is not there. So I am going to show you the environment in which we worked, and then I will talk about neural activity in auditory cortex during auditory guided behavior. I'll start with the sensory responses. There is a story there, not the most interesting one, and then I'll go to task-related activity that is not driven by the auditory stimuli, okay, and this is actually where most of the spikes are. So the riff, why riff? Riff is, well, that's a musical term, but we had to give it an acronym, so it's Rat Interactive Foraging Facility. Okay, so this is it. It's 160 centimeters in diameter. Okay, it has poking ports and it has loudspeakers to which we can play sounds, and it has food and liquid dispensers. So we can give positive reward to the animals and valves for air paths, so we can give negative reward to the animals. Although in the task I will show you, we didn't have a negative reward at all. We observe the animals through a video camera that covers the whole arena. And this is how this looks like from the point of view of the rat. So these are about 50 centimeters high. Okay, and although the rat is pretty small relative to that, some of the rats really learned how to jump and get out of the reef. So eventually we had to put an electrifying wire around it to make sure that they remain inside. Now, as you can see, the rat is not attached to anything. Okay, so we record from really freely moving rats. Okay, we had a number of rounds of these recording devices. Currently, we use a neurologger. So a neurologger means that we have the amplifiers and the digitization of the data on the head of the animal, and then it's written on a memory card, which is on the head of the animal. And we put the rats in, they do what they do for the duration of the session that may last an hour or 12 hours under some conditions. And then we take the card out and read all the data and see what happened. In the data that I will show you here, we recorded with two shunt electrodes that have 32 contacts. In the current projects, we record with NeuroPXI electrodes, so we get 400 contacts inside from which we record activity during behavior. Now, with the 32 channel silicon probes that we have and with the rat logger, we get about 10 good separated enough units per session. An important aspect of the RIF, we wanted to have a system in which there's no manual, very little manual intervention. So we have, at the heart of the technology, there's a computer program that identify the rat. Rat interacts with the arena, means that it moves or pokes or does something, okay? Through the camera, we register the interaction, and then we have a state machine that is in the computer program that reacts to what the rat did and does something. So it can present sound, it can deliver reward and punishments, it can change its own internal state, of course, and this goes on and on. Okay, this loop currently runs at the video rate, which is 30 frames per second. So the main loop, every 30 milliseconds, basically, there's one step of this, we run one step of this state machine. Even with MATLAB and no optimization, this thing can run at 100 hertz. Okay, that's not, with today's hardware, there's no problem of running this even faster. The result is an extremely detailed documentation of what happened. So first of all, we have all the observable events. Okay, so the nose pokes, food pellets, or water or other liquids that we provide to the animal, air puffs and so on. Okay, then we have the internal state of the state machine, okay, that are also coded and marked at the temporal resolution of the system. Okay, we have of course the neural recordings. Okay, the neural recordings are at the highest sampling rate, so they really provide the upper limit of the resolution. And then we have a lot of analog sensors, the most important of which I will show you data is a microphone, okay, on the head of the animal. And so we know roughly what the animal was hearing, and they produce a lot of noise. Okay, so this turns out to be important. And of course, we have the video frames from which we can extract, I will show you a movie in a moment, we can extract information about the pose. With a single camera, you can't get much information, but we do what can be extracted is used. And this whole thing is collected over here. You see about 20 seconds worth of data. And then here it is about 7 minutes worth of data and 2 hours worth of data. I mean, we have this, we can collect this basically forever. We need to change the batteries. The limiting thing are the batteries, and they can operate for 14 hours with this system. With the NeuroPXI, we cannot record so long, but with these 32 channel electrodes, we can basically record 24/7. If we would have wanted, we could have recorded 24/7. Okay, we never did. And here's this is one result of one of our deep neural networks that we use for data analysis. So this is a version of ResNet that identify these 3 points. Okay, so that's the base of the tail and the neck and the tip of the nose. Okay, and from this we get information about the rat heading and the angle between the head and the body. Now, usually when I present the reef to people from at least my community, the reaction is, isn't that too complicated? Okay, so I will let you judge at the end whether we got enough sufficiently interesting data to justify. Frankly, I'm not entirely sure, but that's, you will decide. Okay, so here's one thing that we did in the reef. That was the PhD project of Anna, and Maciek Jankowski is the master experimenter in the lab, so he did a lot of the experimental work, electrode implantation, and developing the paradigms for training the animals and so on and so forth. So this was supposed to be the debugging task for the reef, and turned out into a big project on its own. What the task that we programmed is the following. So the rat moves around in the reef, and if it goes into this central imaginary circle, then the program selects one of the 6, these what we call 6 interaction areas. So a pair of loudspeakers, okay, and play a sound from these 2 loudspeakers, and then the rat has to go toward that port and in order to get the reward. It can leak from either ports. One of them provides food, the other liquid. The rats are food deprived, so they mostly select the food, not the water. And there's no punishment. So if they don't go to these ports, or if they go to a different port, the trial is simply aborted, and they can run to the center and get the next, initiate the next one. A quirk of this task is the fact that we used different sounds for different interaction areas. So we had a lot of discussion which sounds to use, and eventually we decided to use sounds derived from human words. So the students decided to use words meaning that have a spatial meaning in different languages. And these are all acoustically very different from each other. So acoustically, the task is not difficult. What the consequence is that there are really 2 cues for the animal to find the reward location. One of them is the direction, sound direction, and the other one is the sound identity. And there's part of the experiment that I'm not going to describe, where we tested the reliance on sound localization versus sound identity cues. They use both. Sound localization is a much stronger and more natural way of doing it. Okay, but they use also sound identity, and they can be trained to use only sound identity. We play the sound from all the loudspeakers at the same time, and each sound is associated with one location. So they can do this 6-way arbitrary association task too. What you see here is the obligatory movie of rat performing the task. So it runs to the center. It heard one of these sounds. Yeah, we shifted the frequency to higher, the human words-wide frequency. This is why they sound a bit funny. So it runs, it waits a bit, it hears the sound, okay, it runs toward the port, and so on and so forth. That was a mistake. Okay. It turns out that the most important part in the training is actually making them, getting them to remain sufficiently long in the center in order to listen to the sound. Okay, they're very impatient, they like to run, they like to move a lot, and the part of the task in which they had, they needed most, where learning improved a lot their behavior was the part in which they were in the center and they learned to wait a bit. You can sort of see it here. So we had 2 weeks of habituation in which they learned to poke in order to get rewards. The task was not there at all. They just learned that they run to a port, poke, get reward, and then there was a sound to get them used to the fact that reward and sounds are associated. And then what you see is the performance on the first session. Okay, so chance level is 16%, 1 of 6 sports, and already on the first session they did better. Okay, the sound localization task is very natural. They like to be in the reef. It's all very friendly and very easy. So they started above chance, but they never reached 100%. So we don't know exactly what causes them to miss trials. They run to the center, they hear a sound, and then they don't do anything. Okay, we don't quite know why they do this, and they work for us as long as they are hungry. So you see it here. The continuous lines are the amount of food pellets that they got during a session, and this is fixed from day 1 to day 60 to day 180, where we are now with some of these rats. Okay, what changes during training is the number of trials that they needed to do in order to get that amount of food. So they basically, the sessions are long enough so they eat up to satiety. Okay, so these are the behavioral aspects of the task, and I really want to tell you about the neural activity because that was the reason that we did this, we went into this adventure. So I want to start by talking about the sensory responses. We collected sensory responses during behavior and also in what we call passive listening. Basically, we put the rats into this bucket. Okay, and they were in the bucket for about 20 minutes, and we recorded responses to a set of sounds that included also the sounds used during the behavior, the 6 sounds that we used for the behavior, but additional sounds as well. So here's an example. These are 300 and something neurons recorded over a few tens of sessions from one of these rats in response to broadband noise. Now, I want you to pay attention to a few things. First of all, time scales. So the noise burst is 200 milliseconds. It's bounded by these red lines. I show you a half a second worth of data. Okay, these are average responses for all the noise bursts from all levels. Secondly, the latency. Okay, the response occurs within 10-20 milliseconds after sound onset, and that's what happens in auditory cortex. Okay, this is just mean that we put the electrodes in the right place. The third thing is that the response is largely an transient onset response. This looks exactly like the responses of anesthetized animals. Okay, there's nothing special here. Okay, the thing that's special here is this. Okay, so there's also offset responses in some of these neurons. Okay, this is something that we observe rarely with the anesthesia that we usually use. Okay, but otherwise, if you would just show me, okay, the responses during the sounds, I would be hard pressed to tell you whether this is awake or anesthetized animal. We also recorded responses to pure tones. This is something that you do always when you record from auditory cortex. You know, the primary auditory cortex has neurons that are narrowly tuned to frequency. So you want to know that. That's one of the basic response properties of neurons. So what you see here are what we call frequency response areas. On the x-axis, we have frequency. This goes from 1 kilohertz to 64 kilohertz. And these are sound levels. So 10 dB attenuation is quite loud. Okay, 60 dB attenuation is about, I don't know, whisper level in our system. And the color tells you how many spikes occurred. The temporal patterns here are also onset responses, the way that I showed you for the Broadband noise. So we have neurons that are extremely narrowly tuned in frequency and whose tuning width essentially doesn't change as we increase sound level. These neurons are called these frequency response areas are called I-shaped. Okay, these are response areas whose width increases with sound levels, so they are called V-shaped. Okay, then we have things like this where we have significant responses to tones, but they are all over the place. So these are funny shaped, okay, and these are wideband and non-monotonic. There's a wide variety of these tuning curves. Okay, the classical ones that you find in the books are the V-shaped, but the fact that all of these shapes are found in auditory cortex is old and has been shown also in anesthetized animals. So there's nothing new here either. Okay, so sound responses in passive conditions are largely the same boring set onset responses. Okay, there's a rich set of frequency response area shapes, but that's not new. And there are offset responses that are rare under anesthesia. They're actually important. Okay, we use them to, we have now a paper that we hope will be accepted soon after a number of review rounds that discuss both these offset responses and the fact that they seem to be associated with omission responses. So there's interesting stuff there. Okay, but again, let's go to the task relevant stimuli now. We hoped to see interesting stuff in the responses to the task relevant stimuli. So sustained responses, memory related effects, maybe ignition of the global workspace and so on and so forth. Okay. And so we recorded these responses. What you can see here is, this is one of the standard things. We counted how many of these stimuli each neuron respond to. So basically, when you do that, you find that about 2/3 or 3/4 of your neurons respond to any stimulus family that you will select. That's true also for these word-like sounds. And then you have a moderate level of selectivity. There are more neurons that respond only to one or two of these sounds than neurons that respond to all of them, and the pattern is essentially the same in active and passive recording. And so here are the responses themselves. These are the oscillograms of the 6 sounds. Again, note the time scales. This is each one of these is 200 milliseconds long, and I show you here about half a second worth of data. Now, what you see here is the average responses of all the neurons we recorded from one of these rats. Red is one of the conditions, okay, and blue is the other condition, active and passive. What do you think? Which one is the active and which one is the passive? So the answer is the red line show you the responses of the neurons in the passive condition and blue in the active. Okay, now if you look, there are, for me, there's 2 impressive things here. One of them is the responses are smaller in the active condition than in the passive condition. This is something, by the way, that turns out that Brice Batelier observes now in his mice. Okay, so that's and also note that the red responses are essentially, the temporal pattern of the firing is very similar across all of these stimuli. Okay, whereas the blue patterns, they're poor, not very large. Sometimes they don't, for this mouse, for example, for some reason, this sound is not evoke much response at all, but they do vary from one stimulus to another. Okay, so the response in the active condition, both of these are awake, okay, but the response in the active condition seems to be more better suited for making decisions. Okay, they are more different from each other. Okay, so but the crucial thing is that there were different response patterns in passive and active listening. Okay, now it turns out that this is associated with another difference in spontaneous, in ongoing activity. So what we have here, each dot is one neuron, and you see its ongoing activity just before sound onset in the active condition and in the passive condition. So the ongoing activity, the background over which the responses occurred was higher in the active condition than in the passive condition. The responses themselves were really not correlated between the active and passive conditions, but they were tended to be larger in the passive condition. Okay, that's the quantification. I'll get back to this issue of spontaneous activity at the end, ongoing activity, but that's the conclusion. Okay, responses during task performance differed from the response in passive listening. Okay, they were more distinct, so they carried more task relevant information, but they were well, we found them boring. And the comment I want to make is that the higher ongoing rate in the active state by itself may cause the weaker and more distinct responses. And this is that we actually can show in a model. Okay, we have a standard model of auditory cortex that we published a few years back. And if you actually increase spontaneous rate in that model by 20%, which is what happens here experimentally, we see these effects. The reason is that more activity goes into cortex, there's more synaptic depression, and the more synaptic depression is the thing that actually reduces the responses. So this brings up the question of what is the origin of the higher ongoing firing rate. And this, I will show you in a moment that we have a handle on that. Okay, but these sound-driven responses are actually only a small part of what's going on in auditory cortex during behavior, and partially it's our fault. Okay, so I want to talk a little bit about what happens when we don't present sounds. In the task, sounds occupy less than 10% of trial time. The reason is that we present these sounds once every 2 seconds. The duration is 200 milliseconds. Okay, and they are presented only when the rat is actually performing the task. So more than 90% of the recording session, okay, the rat doesn't hear sounds that we presented. The responses to the sound relevant stimuli are short and transient. This is what I showed you. They don't outlast the stimulus by much. Okay, but the neurons are still firing. Okay, I showed you that they have an ongoing firing rate. So the question is, what are the neurons doing during the other 90% plus of the time? And in order to do that, we did something that at some point, okay, we did something we never did before, and this is opening the time scale. So instead of looking over half a second worth of data, okay, I'm showing you now firing rate of a neuron recorded in one of these experiments over 30 seconds, okay, 30 seconds of time. Okay, that's not a mistake, okay, these are seconds. So first of all, these are 30 seconds, okay? The gray line is the time that the rat crossed this imaginary line into the center of the arena. As far as we are concerned, that's start of trial. In terms of the rat, start of trial occurred a little bit before, but we don't have a handle on that, okay? We only know when the rat crossed to the center. Okay, a short time later, I really put the red line only I shifted it by a little bit so that visually you will see that they are different. That's 200 to 500 milliseconds later. On this time scale, that's essentially imperceptible. There's a sound. Okay, and then what you see in the green and magenta lines are other traces of things that we know happened during the trial. So the green line show you the running speed of the animal. The animal, what you see the animal do is accelerating when it runs toward the center, then decelerating as it slows down in order to listen to the sound, then accelerating again towards the port of reward, and then decelerating when it reaches the port. Okay, so basically this is roughly the average moment in time in which the rat initiated the trial, and we don't know exactly where this time is. And the magenta shows a trace of the power, sound power in the microphone on the head of the animal. So when the animal is running toward the center, it's actually, there's a reduction in the self-generated noises. Okay, this is an increase in the noise that's due to the fact that the animal is got to the wall, it hits a little bit the head on the wall, and then it eats the food pellets. So all of that creates noise. And this noise peak is actually the same thing from the previous trial. Okay, so now on top of that, with the black line, you see the firing rate of the neuron. Okay, so the thing that I found still impressive after looking many times on these things is this huge increase in firing rate. This event happens over about a second or so. Okay, and it is really large. It is about 3 spikes per tile. Okay, so this represents a lot of spiking. So much spike. The sound response of this unit occurred somewhere here. Okay, but at this time scale, and you really can't see it. Okay, it's smoothed over because of the coarse time scale that we use for representing the changes in firing rates. Okay, now to show you how large it is, this is a raster plot of this unit. So what you see are 500 and something tiles. Close to the center crossing here, you can see these additional spikes on this single tile plot. Okay, the tiles are arranged by time to the next tile. So this blue line represents the times of the following tile, and that you see the same, this line is basically the same one as that one, okay, shifted by one tile. Okay, here's another unit. Okay, for this unit, you actually can see the sound response. It's this bump. Okay, so we count spikes. This is about 10 spikes per tile in this case, and this is about 1 spike per tile, and this is typical. Yeah, so these things are actually, these events, okay, are actually 10 times larger than the sound response. Okay, and more examples. So what is going on? This is going on. Okay, what I show you here are, again, 450 something neurons recorded from one of these rats. This is plus minus 5 seconds. And here I organized these, I arranged these neurons by the time of this peak. Okay, now this is two comments on this. First of all, some of these neurons respond to sound. So these are, for example, the response to broadband noise of the same neurons. So you see again these short latency offset responses and some off responses. Okay, so whether this, some of these bumps occur with and without sound responses. Secondly, this is results from 4 rats, and here we computed ordered the units by the average responses to all the odd trials, and here you see the same neurons ordered the same way, but the average of the even trials. Okay, so this ordering, the fact that these peaks seems to occurs during the trial and to span the duration of the trial seems to be well, to at least withstand this type of control. Now, what accounts for these neural responses? So very shortly, we try to account for this modulation with everything that we measure in the reef, the microphone signal, velocity, location, and direction, and so on and so forth. and also with a null model, okay, in which firing rate depended only on the current time relative to sound to the tile onset. It's not sound onset, tile onset. Okay, and it turns out that about 1/3 of the units are best accounted by this time model. Okay, so sound, velocity, and all of these things did not account well for the responses, and the only thing that account for the fact that they fire at a certain time during the trial, okay, is assuming that they are really sensitive to the time in the trial. Okay, about 1/3 of the neurons were best described by sounds, so mostly sound, self-generated sounds. So this is good because we are in auditory cortex. A lot of sensitivity to velocity that was known before, and then location, head direction, all kind of other stuff. So what I showed you is that many neurons had large slow firing rate fluctuation log to trial time. And auditory responses are often small and lying on top of these large slow firing rate fluctuations. And in fact, these fluctuations account, not only can, okay, they accounted for many different trial-related variables, largely, I mean, with a plurality of neurons that seem to track time in trial. Okay, so last point is that the large task-locked firing rate modulation that I just showed you also account for the differences in response rate in the ongoing rates. So they seem to be also, they could be the source of the differences between responses in the active and passive conditions. So this is it, okay? What I showed you is that conclusion, okay, is that the task-related activity that is not driven by auditory stimuli is actually most of the spikes that we see during trials, about 10 times more than sound responses. Activity in the auditory cortex is much more than just the responses to sounds, okay? We have these time encoding neurons that seem to be a new family of neurons that we didn't know and that we before, and that seems to be a large population in the cortex. Okay, so where do we go from here? First of all, we have the current mess that we still need to account for. What are the mechanisms that produce these responses? Are they inside the auditory cortex or not? Are they results of training? And what are they for? Okay, one thing that we know is that they make the sensory responses even more boring than in the passive case. We are trying to look for more links with behavior, so since we have this very fine-grained analysis data about the behavior, we develop fine-grained methods in order to analyze and grade the behavior and link the neural activity with the behavior. And of course, I mean, this is technology of 5-6 years ago, so we would like to have just whet our appetite. We need more documentation of the behavior. Was it worth the trouble? I leave you to answer that. I leave to you the question. Here are again the students. Okay, they are smiling because they are happy in Jerusalem. And we have a lot of good labs. The younger people here, I strongly urge you to look for positions at LSEC. We have just came out with a new call for postdocs in the lab supported by the center, and thank you for listening.